Hi, everybody. This is Dr. Launderville. This is online podcast number 7. Online podcast number 8 will be your last one before the final. So these are just 2 more podcasts before the final that'll help you hopefully prep for the final. But it's new material. I want to mention that the Biology Undergrad Research Symposium is going to happen after finals on May 13th. So any of you that are doing undergrad research that is biology related, whether or not you're a bio major, you can participate. And the way we're going to do it is we're going to do it on Twitter. There's going to be judges from biology. There'll be cash prizes, $150 for first place. So far, not many people have signed up, so your chances are good. Send me an email if you're interested. It's a good little thing to have on your CV. Okay, so these last 2 podcasts are going to be focused on cancer. I'll say, as I've said many times, the stuff that we've talked about in this class has set you up for understanding the cellular and molecular basis of cancer as we understand it as of 2020. The idea of finding a cure for cancer is not as crazy as it used to be, but it is a really tall sort of, really complex sort of problem to solve, because cancer is just a common phenotype, that being uncontrolled cell growth, and there are many ways that cells can get to that phenotype. So they could resist cell death, resist apoptosis. They could have sustained proliferative signaling. So the growth factors that need to arrive and bind to those RTK channels, the cells can make their own growth factors and have those bind to their own RTK signals. They can evade growth suppressors. So some of the pathways we've talked about have a default where the cell will grow if it doesn't receive a signal. Some have a default that will not grow if it doesn't receive, or if it does or doesn't receive a signal, and it can get around those pathways. We'll talk more about in these lectures how they activate breakdown of the protein matrix around cells that allows cells to move from one place to another and metastasize, cause a cancer away from the original source of the cancer. There's a phenomenon that we'll talk about in these 2 podcasts where most cells go through about 50 cell divisions and then they stop. And cells can get past that sort of limitation and just keep dividing. And another thing is tumors have a general problem in that they grow so fast, they grow faster than their blood supply, and you need to feed those cells with oxygen and get waste materials away. One way you can get around that problem is just induce chemical signals so you grow your own capillaries. It's called angiogenesis. So there's so many different ways that if things don't work the way they're quote-unquote supposed to work in a wild-type way, the default is you end up with uncontrolled cell growth. And so trying to find a fix is whack-a-mole at a very big scale. There's all kinds of different ways that cells can evade any kind of fix. So it's amazing it ever works. And the statistics are that you're going to know someone, if you don't already, who's been affected by cancer. a third of all humans in the U.S. are going to be diagnosed with cancer at some point during their life, according to the American Cancer Society. You, as a result of taking this class, have some skills, and you can understand this language better than a lay person who is not a scientist. You can understand a lot of the vocabulary that are in these papers about cancer, are in these papers about clinical trials, and you should use that to help people, at least offer them that help. So I've had the experience of knowing, unfortunately, a lot of people in my life, people I've connected with that have been diagnosed with cancer, and they're scared, undoubtedly, as anybody would be. And when I've offered to them, helping them interpret these papers, helping them interpret clinical trials, helping them know what the mechanism is for the drug that they're on, that their oncologist is putting them on, helping them understand what is happening to them and why it's happening to them. For people who want that help, it's a big comfort. Now, some people don't want that help. They get this kind of diagnosis, they just don't want to know, and that's their business. It's their cancer, it's their life to do with what they want. But for people who do want to know, they don't typically get the kind of face time that they need from their oncologist because doctors are busy. And you can help them figure that stuff out so they can advocate for their own care better, so they know what's happening in their bodies and they feel more in control. And that's a real gift for people who are going through this horrible thing. So consider about offering it up. Now, I would not say you should extend that knowledge to the point of offering advice, because none of you are MDs yet. But you can extend that in order so that people can ask good questions and help them interpret papers. And usually physicians will respond really well to a well-educated patient, and they end up talking to them differently if they know that they are reading a lot. So there it is. Okay, here's an H5P question for you. All right, so just going back to understanding mitosis, and most of what we're talking about is mitosis and not meiosis, so cell division, prophase, metaphase, anaphase, telophase, and that's all part of mitosis. And then interphase would be these growth phases, first growth phase and second growth phase, and then a synthesis phase where you double your DNA. And cell is going to make decisions about how to go through these different phases. So how does a cell go through the different phases of mitosis? How does it decide, quote unquote, I'm going to go into anaphase now? How does it decide that it's going to hang out in the first growth phase and not divide? A lot of your terminally differentiated cells do that, and some may hang out like that for years and not divide. And then when they do decide to divide, how do they make that decision? And then usually once the decision is made to enter S, the cell will complete the cell cycle. But there are checkpoints along these different parts of cell cycle that are, not surprisingly, important in understanding cancer. So when cell biologists were first trying to figure this out, everything was in the realm of factors. Remember, factor is what a biochemist or cell biologist calls something when they don't know what it is. They just know something in this solution is causing something to happen. So what they would do is they take cells and they would fuse them with another cell and see what happens. So you take a cell that's undergoing mitosis and you fuse it with another cell that is not undergoing mitosis and see what happens. So if you take cells that are in G1 and you fuse it with another cell, you'll get some condensation of chromosomes, but they don't associate it as centromeres, so they don't look like this sort of thing that you typically see, say, during mitosis. If you take cells that have been through G1S, and now they're in G2, and you fuse that with another cell, those chromosomes condense more, and sometimes you get this homologous chromosomes associating, sometimes not. No matter what you do, if you fuse one cell with another cell, you get the nuclear envelope dissolves. And then some more sophisticated experiments. They took some frog eggs that are in G2, treat them with progesterone, and then they start going through meiosis. So I said most of what we're talking about is mitosis. This is just meiosis because it's an egg, but it's still cell division. If they take that stimulated cell, okay, it's been stimulated with progesterone, and it starts to go through meiosis, and they take some cytoplasm out of that cell, and then they add that to an unstimulated cell, hasn't been treated with progesterone. Same thing happens, and then it starts to go through meiosis. And you can do that multiple times, is that you take this cell, take the cytoplasm of the cell, add it to an unstimulated cell that has not been stimulated with progesterone, and it'll start to go through meiosis. And you can do that so many times that if you calculate how much the factor has been diluted from the original cell, if there was some magical factor in that original cell coming from the progesterone, it can't be leftover progesterone that is causing it. So they said that whatever this mysterious thing is that gets turned on by progesterone, we're going to call it a maturation promoting factor because these cells are maturing, MPF. And if you track the activity of the MPF over a cell cycle, its activity changes with the cell cycle. So its activity changes over the course of G1, over the course of S, over the course of G2, and over the course of mitosis. And this activity also still cycles even if you take the nucleus out of the egg. So that if you take the nucleus out of the egg, they are not transcribing any new genes. They're just relying on whatever mRNA was in that egg when you started this experiment. Not surprisingly, because everything is a kinase, MPF is a kinase. And in order to get the kinase activity, you need protein synthesis. So chemically you can stop protein synthesis when you do these experiments. And if you do that, you don't get any more MPF activity. So it's a kinase that requires protein synthesis for it to be active. The protein that gets synthesized is a protein called cyclin. It's first discovered in sea urchin embryos, which is just another model cell system, as giant eggs, giant cells that you can play with. And they found that that's the protein that gets made, that this kinase depends on, and the kinase is called a CDK, or a cyclin-dependent kinase. So, if we measure the concentration of cyclin, so cyclin is the thing that is allowing the kinase to happen. It is not the kinase itself. It's just a protein that allows kinase activity to happen. So if we track the concentration of this protein that allows kinase activity to happen, it goes up and down with the cell cycle. But if you add RNase to these cells, you get no cyclin. Okay, so cyclin is present as an mRNA in these eggs and is not being transcribed anew by these eggs, because you take the nucleus out of the eggs this you still get activity. But cyclin mRNA has to be there, and the cyclin actually gets turned into protein during the cell cycle. And then it's destroyed, and then turned into protein, and then destroyed. Okay? If you make if you treat it with RNA, so there's no cyclin, and then you add just back in a cyclin that can't be degraded, okay? You get an increase in activity, but then it doesn't decrease. So there are specific cyclins that have mRNAs present that are made, the protein is made, and then you get that activity, and then that protein and its mRNA are destroyed, and then a new cyclin is made, and then destroyed, and then a new cyclin is made and then destroyed. Okay? Okay, here's another H5P question for you. So, what is a cyclin-dependent kinase? It's one of those rare things in cell physiology where it's exactly what it says. So there's a kinase domain and a cyclin binding domain. Cyclin is the protein that binds to the kinase and turns the kinase on. It is a kinase that is cyclin dependent. Okay? They have to bind cyclin to be active. There's all kinds of different cyclin dependent kinases. Some require phosphorylation to be activated. Cyclin activating kinases, and some are regulated by inhibitors called cyclin dependent kinase inhibitors. But there's lots and lots of different ones. And here's a table of all kinds of different ones. And then there are phosphatases that go with them. So kinases to add the phosphate, and then cyclin-dependent phosphatases that are to remove the phosphate. Okay, these different kinds of cyclins are present and active in different parts of the cell cycle. So you have cyclin-dependent kinases that are present and active during the G1S phase transition. And then you have S phase cyclin-dependent kinases, and then you have mitotic cyclin-dependent kinases. There are lots of different cyclin-dependent kinases, and they, for normal wild-type cell function, go ahead and divide your cell, and then you stop just like you want to. It's imperative that the right kind of cyclin is made at the right part of the cell cycle, and then it is destroyed, that it does not stay around for a long time. So cyclin-dependent kinases that stay active too long, are not destroyed, in other words, can lead to uncontrolled cell growth because they will bypass these checkpoints. Okay? So, speaking of checkpoints, there are places where the cell will stop and inventory what has happened in its past history, and if what has happened in its past history is something bad, something that the cell would not want to replicate in a new cell, then it will try to fix that problem if it's fixable, and if it's not fixable, it'll activate apoptosis and kill off that cell in those cell cycle checkpoints. And not surprisingly, these cell cycle checkpoints are some of the most common oncogenes. So an oncogene is a gene that you need it to work normally for normal cell function, but it also commonly breaks, and if it does break, causes cancer. So earlier I gave an analogy of if you're driving a beater car, and for a given car, a particular thing commonly breaks, and that will cause the car to no longer function. You need that thing to work, but then it's also commonly breaks. So for example, my wife has a 1995 Jeep Wrangler. It's an old car. Jeep Wranglers are famous for having a frame that rusts out. And actually it's our second one, and the first one, the frame did rust out. So you got to have a car frame. Very hard to go down the road if you don't have a car frame. And so you need that to be a car, a functioning car. But also that's a part of that model of car that tends to break. And so that would be equivalent to an oncogene. So here are some oncogenes. We're going to talk about a bunch of different these, and these are involved in cell cycle checkpoints. So these genes here are involved in checking for DNA damage that may have occurred during G1. So some cells will hang out in G1 for most of their life, in fact years. They may have acquired DNA damage from UV radiation or exposure to free oxygen radicals or whatever it may be, and these genes are involved in monitoring for that damage, and then also trying to fix that damage. So, if those genes break, that means that it could be that you, at the point where you're starting to duplicate your DNA, you would be duplicating damaged DNA, and that would lead to perhaps uncontrolled cell growth. Okay. There are genes that monitor how well did the DNA duplication process go when you're going from one copy of your chromosomes to two copies of your chromosomes. How well did that go? It's possible that there's been a big mistake in there, and then these things would check for that. If these are mutated and you don't get that check, it's possible that a cell that has made a big mistake in its DNA duplication will go ahead and duplicate, and you don't want that to happen. So these checkpoints are really important for cells. The typical response at a checkpoint is to check, make sure nothing disastrous happen. If it's a disaster that can be fixed, to call in the biochemical mechanisms that'll fix that disaster. If it can't be fixed, then the default pathway is kill that cell. If any of those things happen, where the process to check on the cell breaks down because there's been a mutation or some kind of problem, then you have cells being duplicated that you don't want duplicated. And those cells can evade the normal checkpoints, and so they're evading the normal decisions on whether or not to duplicate cells, and therefore you have uncontrolled cell growth, uncontrolled cell duplication, cancer. Okay, here's another H5P question for you. Okay. When cells are in the G1 phase, the first growth phase, they're going to make a decision on whether or not to start to divide, the S phase. Okay, usually once they enter S, they go the rest of the way through G2 and then through mitosis. So the G1 S transition is really important. In cells, there are various mechanisms that cells can use to monitor their environment. So there's a protein called CLN3 that has activity that is tied to nutrients that is in yeast. In vertebrates like you and me, there's a cyclin-dependent kinase 4 and 6 activity that is tied to growth factors. There's also mTOR that we talked about that is monitoring the environment of the cell and say, are there nutrients available? Should I grow or not? Should I duplicate or not? Once you get over what these monitoring system says go ahead, you should grow. Then the cells will phosphorylate this protein called RB. Okay, and RB is normally inhibiting a transcription factor called E2F, and E2F is a transcription factor that is going to turn on genes that are required for the S phase. These cyclins that are active during the S phase that will activate cyclin-dependent kinases that'll turn on the genes that are important for duplicating your DNA. Normally, you want that off because the cell is sitting in G1 and it's growing. It doesn't want to duplicate its DNA. If you phosphorylate RB, then it comes off of E2F, and now E2F has permission to go ahead and make mRNA for those proteins. And so these cyclins will be synthesized, cyclin-dependent kinases become active, and the cell will enter the S phase. Okay. The gene product RB is inactivated in almost all cancers. So almost all cancers have non-functional RB in that E2F is not inhibited. It is constitutively making the genes to synthesize DNA. And so cancer cells will just constantly get a signal to say, go ahead, duplicate your DNA, and start replication. Okay. Other things where cyclins are pushing through cells through the cell cycle. So early in this podcast, we talked about those early experiments where you fuse two cells together, and sometimes chromosomes condense, sometimes they don't, but no matter what, the nuclear envelope disappears. So how does that happen? These cyclin-dependent kinases have targets that are lots of different proteins that are not just other cyclins. Some of these proteins are the physically holding the chromosomes together, or physically giving the nucleus its shape. Like the lamins that make up the nuclear envelope have this cross-structure sort of network. They're bound together. They're made of these tetramers. If they get phosphorylated by a maturation promoting factor, which is just a cyclin-dependent kinase, then they depolymerize and the nuclear envelope comes apart. And then after you've been through telophase in mitosis, then you have phosphatase that removes these phosphates and they repolymerize and reform a nucleus. Another thing that can happen to move things through the cell cycle, it's specifically in mitosis part of the cell cycle, there's a metaphase anaphase transition. So metaphase is where the chromosomes line up in the middle of the cell, remember, and then in anaphase you have one chromosome with two chromatids per chromosome becoming two separate chromosomes, each one with one chromatid. The two chromosomes per chromatid are held together by this protein complex here. And during that metaphase-anaphase transition, you've got to break apart this protein complex so that you can take that two chromatid chromosome into two separate chromosomes. There is a protein complex called the anaphase promoting complex, or APC. And it is, look at that, a ubiquitin ligase. Ubiquitin is everywhere. So it's going to add ubiquitins onto stuff. In this case, it's going to add it onto a protein called securin, which is an inhibitor of a protease called separase. So the anaphase promoting complex gets activated by a CDK, and I'm pretty sure that's correct. And then once it's activated, it polyubiquitinates securin. Securin gets degraded in the proteasome. That releases separase. Separase is going to cut the link that is holding these two chromatids together, and now they can go through anaphase where 2 chromatids per chromosome becomes 2 separate chromosomes. CDC20 is the specificity factor on this anaphase transition, anaphase promoting complex. Say, go ahead and tag securin and not any old thing. And separase is related to a caspase, caspases that we saw in apoptosis, but it's a protease that's going to make this cut here. So this is promoting the movement from metaphase into anaphase. Okay. We mentioned that the lamins get phosphorylated to come apart, and then they get dephosphorylated to come back together and reform the nuclear envelope. That's the protein part, but then there's also the membrane part of the nuclear envelope. And during telophase, the vesicles that are the lipid part of the nuclear envelope will associate themselves with single chromosomes, and they form a thing called a carriomere, and then these carriomers fuse to form the nucleus, and the new nuclear envelope gets set up by these lipid vesicles fusing together, and then they start importing things like nuclear pore complexes, and then the lamins will set up the shape of the nucleus. Okay, on a more macro scale, cancer is abnormal cell growth, growth that is not under normal control. So it has evaded those checkpoints, and it is blasting through those checkpoints and saying, I'm going to divide no matter what. You've heard these terms, benign and malignant. Benign tumors are self-limiting in that they have contact inhibition. When one cell bumps up against another cell, it'll act as a signal to stop growing. But malignant tumors can overcome that contact inhibition, so they can grow on top of each other. They can also become mobile, so that they are more amoeba-like, like a macrophage, and then they can extrude processes in the cell and actually move from one place to another. They can secrete proteases that will digest the extracellular matrix. So if they are if it's a cell that's surrounded by extracellular matrix, it'll start to move and dissolve or depolymerize the extracellular matrix around it, and then eventually get into some kind of fluid tissue like lymph or blood, where it can travel far to another part of the body. In a gross section, tumors look like this. The whitish part is much less vascularized than this part here. I think this is a liver tumor. On a slide, what you're looking for is a great density of nuclei. So cells aren't under normal cell control. They're not monitoring their environment for nutrients in the same way. They're getting a signal to go ahead and divide more often than a normal cell would, and therefore the ratio of cytoplasm to nuclei is different in that there's a lot more nuclei per cytoplasm. A lot of cancer cells are just like a little thin strip of cytoplasm around a nucleus because they're constantly dividing and no time to grow. So if you treat a section of cells with a nuclear stain, this dark purple stain here, you see great density of nuclei in this area and much less density of nuclei in this area. So this would be considered the tumor cell, this would be considered the normal cell, and this would be considered the margin. So in something like a skin cancer, they would take sections of the skin cancer while the patient is there, while they're doing the surgery. They would prepare it and look at the cells under the scope and look for the presence of tumor cells until they get to a slice of tissue that is all healthy tissue, and then they stop. So that's still a lot, even with all our molecular tools, a lot of cancer diagnosis and typing is done by histology. Okay. As I just mentioned, cancer cells have very high DNA to cytoplasm ratios because they are not under normal cell cycle control. They are constantly dividing. They may have several nucleoli in the nucleus because they're pumping out all kinds of RNA, and they may have abnormal numbers of chromosomes. So instead of the 23 pairs that would be in a normal somatic cell in a human, they may have many more than that, or an uneven number, because that's part of cell cycle control, is just counting how many chromosomes you have, and if you're not paying attention to that, you have an unusual number of chromosomes. In terms of differentiating malignant cells from benign cells, malignant cells tend to be more like a stem cell in that they are less differentiated than a benign cell is. So it can be hard, if you're just looking at a single cell, to know necessarily that this came from a liver cell or this came from a heart cell if it's a malignant cell because of this. But then there are some extracellular markers that you can usually use to identify original source of a cancer if you're looking at a malignant tumor. So benign tumors, because they have contact inhibition, they are uncontrolled cell growth, but they have a little bit of control in that they have contact inhibition. And so they haven't gone to cells becoming mobile yet. They haven't gone to the part where they're destroying the extracellular matrix and trying to get to a fluid tissue. So if a benign tumor is caught and removed, it's not a threat for developing uncontrolled cancer, except it may be its physical presence may be a threat, like maybe it's pushing on a nerve or something like that. However, benign tumors can become malignant in sort of a developmental pathway, and there's some cancers where it's really well characterized how benign tumors become malignant if not treated early. As I said, you can have unusual number of chromosomes in cancer cells. So here's a normal karyotype. These are chromosomes that have been painted with fluorescent dyes. They're not normally this color, but this is pair 1, pair 2, and so on in a human, whereas in a cancer cell karyotype, you can have all kinds of unusual chromosome numbers, and you can see it's not that you have 3 of every kind. Some chromosomes you have a normal complement, some you're triploid, some you're quadruploid. It can be all over the place. More about metastasis. So when a benign tumor becomes mobile, it's called metastasizing, and these cells that move from the original source of the cancer can cause cancer at a remote site. In fact, this was first figured out in some really horrible experiments where they injected prisoners with cancer cells and then induced cancers at the injection site. They told them they were testing them for cancer, but instead they were giving them cancer. That was done in the 70s. So in a metastatic cancer cell, they're going to turn up proteases that are going to break down extracellular matrix. They're going to down-regulate cell adhesion molecules. That is how a cell knows what cells are next to it and what kind of cell it is. So it'll stick to other cells around it, and it says, okay, I'm a hepatocyte, I know what to do. These cells that are migrating out of the tumor will eventually migrate their way into the bloodstream or the lymph, and most of them won't survive. So either they are missing what they need to stay alive, or they get found out by a macrophage or natural killer cell and they die. Only 1 in 10,000 would live, but then if they do live, they can establish themselves on a remote site and where they can cause a new tumor. So when someone is diagnosed with a tumor, they will usually do some imaging and look at lymph nodes and also look at the lungs, because the lymph nodes are collecting cells from the body. As we learned in the lecture on immunology, those cells that are metastatic can collect in the lymph nodes and start a new cancer there. And so that's one of the first places to look. And also in lungs, for the lungs to do their job, in that to exchange blood gases, you have to have a very thin connection between the outside oxygen atmosphere and blood cells, and so you have a very tight fit as cells are going through those vasculature in the lungs. And so often these metastatic cells can get stuck in the lungs and start a new cancer there. Some of the effort now in terms of diagnostic tests is looking for cell markers on metastatic cells, and so that you could routinely monitor somebody from a blood sample and say, do you have any cell markers on the outside of the cells from the sample that could indicate a metastatic metastatic cancer, and then we'll go looking for it. And there's some of the possibilities look really promising for that kind of test. There is an idea of a cancer stem cell. This was pretty controversial at first, but not so controversial now. So the idea is that in a tumor, which is cancerous, not all the cells in the tumor have the same characteristics, and there's a range of cells in the tumor in terms of how differentiated those cells are. How much are they like a liver cell or not? If it's or just a general progenitor cell, if we're talking about a liver tumor, for example. And the more differentiated end of those cells, they don't divide, or at least don't divide as often. And cancers need to keep dividing. So if you could identify the populations of cells within a tumor that are more likely to divide, those are the ones that are more likely to give rise to new cancers, become metastatic. And if there's something about those cells that makes them more likely to divide, could we possibly inhibit that and therefore inhibit the growth of tumors? So there is now, like I said, pretty well accepted that there are cells on the less differentiated end of things in a tumor that are referred to as cancer stem cells. There are many different factors, protein, non-protein, that are being investigated as to, will this tell me you are a cancer stem cell? If you have this thing on you, are you a cancer stem cell? And then maybe I could develop a targeted antibody against that thing that will kill all the cancer stem cells. One of the candidates is called CD138. It's also called Syndecan. And this is a glycosylated protein that is on the outside of cells, lots of different cells, and specifically bone marrow cells in multiple myeloma, which is a bone marrow cancer. This glycosylated protein binds lots of different molecules, and those include growth factors, and these extracellular domains of these proteins, which are membrane bound, they can cut off their extracellular domain and then throw that into the environment. And so if it's binding growth factors, you can get like a gradient established from this process. So you have these CD138 that are bound to the cell, and they're binding growth factors, so you have a high concentration of growth factor right next to the cell. And then some of those are shed from the cell, and so you have a little less growth factor further away from the cell, and then a little less even further away. And so for something that was looking for stimulation of growth factor, in order to stimulate those cells, you'd have a gradient of growth factor going to the cell that is expressing CD138. It is a candidate for one of these markers of cancer stem cells, and it's been pretty controversial as to, first it was saying, okay, this is it. If we know that you're making a lot of this thing, you are a cancer stem cell. And then a bunch of papers came out and said, nope, that's not it, because we found these other cells that are part of the tumor that are not making this CD138, and they can still induce new tumors. So we think that the ones that aren't expressing it are also cancer stem cells. So there's a paper that just came out, 2020, published in Leukemia, and where they're saying that whether or not you make a lot of this stuff or you don't make much is a switch. And it's a switch for, are you going to stay in place and grow this tumor, or are you going to try to metastasize? So if you make a lot of the CD138, those cells are getting the signal to stay in place as a tumor and grow that tumor. If you're making a low concentration of CD138 on your surface, those are the cells that metastasize the best, and they are the best to establish a new tumor in a new bone someplace else. So it's probably that all the previous studies were all right, and that there's a difference between how these high expressing and low expressing CD138 cells behave, but that both can cause cancer and both can respond to establishing cancer in a different way. And that, according to this paper anyway, the low expressing CD138 cells are the ones that we should go after in terms of stopping the spread of multiple myeloma. Okay. So, another thing about trying to identify cancers and conditions that lead to cancer is that where the tumor is growing makes a big difference. So a large percent of cancers are due to chronic infections. And this seems a little counterintuitive at first, but if you think about it a little bit more, it makes a lot of sense. So if you have an infection, there are, as we're learning now with all this, the COVID-19 research, and you learned during the immunology lecture, that an infection induces inflammation, activates your innate immune system, and will call other cells of the immune system to that site. There'll be macrophages will be attracted to that site as part of the cytokine storm sort of thing. When the infection is taken care of, there are growth factors that are released by the cells that have been damaged. So the cells have been damaged. They're either cleaned up by macrophages or they went through apoptosis and then cleaned up by macrophages, and there are cells missing because they have been damaged and they died. So cells will respond to that by secreting growth factor and saying, we need more cells to fill up this thing that has been injured, and those cells are now gone. So those growth factors get released at the site of the inflammation, and then the extracellular matrix gets reworked. So let's give these cells a place to grow and to attach to, and that will heal your infection or injury. And that works all great if you've got a splinter and you had inflammation at the site of the splinter and you dealt with the infection at the splinter by calling your innate immune system to the cells, to that site, and eating up all the bacteria that were there. And then any cells that are destroyed, you release some growth factor at that site, and you rework your extracellular matrix, and now you're ready to go through mitosis, duplicate some cells, and you'll grow some new skin, and you'll fix that splinter, and a week later, and it's like it never happened. But if this happens all the time, that you have a chronic infection, then you get these sorts of signals all the time, and this is like a recipe for cancer. So let's make cells grow. Let's rework the extracellular matrix. Let's secrete some growth factors. Let's stimulate mitosis. All these things are what cancer does. And so chronic infection like gingivitis, chronic infection in your mouth and your gums. There's a lot of evidence now that can lead to cancers and cancers in other parts of the body. There's some other implications for inflammation in establishing Alzheimer's disease. So doctors will be on you about this if you're on a regular wellness visit, and that they are looking for signs of any kind of chronic infection, because it can have really damaging effects that are far-reaching. And my dentist has all kinds of articles pasted to his lamps and stuff, so that while you're sitting there getting your teeth worked on, you have no choice but to read about how taking care of your teeth can save your heart, for example, or save you from cancer. There are some cancers where the molecular pathway for how these cancers develop is really well worked out. Colon cancer is one of those cancers. So first thing happens is you lose the APC tumor suppressor gene, so anaphase promoting complex. If you're activating that securin and allowing those cells to go from metaphase to anaphase, if you lose that checkpoint, you just go anyway, and you can have unusual chromosome numbers showing up in the downstream cells. Later on, you get activation of KRAS oncogene. Remember RAS, we talked about a lot for the last test. So this is a oncogene that stays on more than it should, or is hard to turn on when it's supposed to turn on. So it's phosphorylating more than it should, or phosphorylating less than it should. And so the result is uncontrolled cell growth. Then you can lose P53. We'll talk a lot about how P53 works in the next podcast. And then you can get changes in the extracellular matrix and can become metastatic. These are polyps. They are considered benign. These are things that you'll have checked starting when you're age 50 in colonoscopies. There may be something less invasive by the time you're 50, but every 5 years or so, if you're starting at age 50, or maybe I think it's every 10 years. I have colon cancer in my family, and so I'm at an increased risk, so it's every 5 years for me. It's not a fun test, but it's really important, because if you can catch polyps and remove them early before they become metastatic, then you're okay. And if you wait too long, then we know the changes that are going to happen, and we know that it'll lead to metastatic cancer. Okay, so in studying cancers, cell biologists study them in culture in addition to in the macro scale in people. In culture is a lot easier than in people in terms of trying out the effectiveness of new drugs. And in culture, some cells will lose normal control of growth and proliferation. So you take cells out of the body and you grow them in culture. They are outside of their normal environment, and you're providing them with growth factors, and some of those cells will acquire mutations and lose control, normal growth and proliferation. And when they do, they change shape. They are less dependent on growth factors, and they lose contact inhibition, so they grow over each other. So normal cells in culture that still have contact inhibition, that have not lost normal cell features, normal checkpoint features, will look like this, and that they are flattened. Cells are not growing on top of each other. In transformed cells, this is called transformation or malignant transformation. It's not the same thing as transformation when you're cloning in bacteria, but it's the same sort of idea, and that the result is a different, genetically different thing than before transformation. So in malignant transformation or transformation of cells, they lose contact inhibition. They grow on top of each other. You can see much more of a 3D sort of situation here. And they don't need as many growth factors, and they can grow forever as long as you keep them fed. So a cell strain under normal conditions of cell cycle control will last about 50 divisions and then will stop. Okay, they're under normal control of cell division. A cell line that has been transformed is immortal. So if you keep it fed and diluted, not too many cells per plate, it'll go on and on and on for as long as we know. And this is for as long as we know since the 1950s, when the first cell lines were established. And those cells will keep on dividing forever, as far as we know. Also in cell culture, you'll see a lot of reference to fibroblasts. These are fibrous germinating cells. A lot of times when you take cells out of culture, they will lose some of the characteristics that they had in their tissue and become fibrous germinating cells, just sort of generic cells. All right, how do you take a cell that has control of its transition from the different parts of the cell cycle to one that is not under normal control? There's lots of things you can do with it, and all these things are the things that cause cancer. So radiation, expression of oncogenes, mutant copies of genes that you need, but when they are mutated, they cause cancer. Chemical exposure to viruses. So yes, viruses can give you cancer. You can actually catch cancer from a virus. They have no contact inhibition. They can grow uncontrollably as long as you keep them fed. In addition, they have increased mobility of integral membrane proteins. So it's not because of the phospholipid, but the connection to the cytoskeleton underneath is altered. And so these integral membrane proteins move more than they would in a wild-type cell. They increase glucose transport. They express a glucose transporter that has a lower Km for glucose. Really good at getting glucose in, and I'll explain why that makes sense in a second. There's a protein called fibronectin that will usually form a meshwork over cells under normal control in culture that is going to be mostly gone in transformed cells. And they do express different genes, but not as much as you would think. So if you do RNA-seq experiment on transformed cells and normal cells of the same cell type, say liver cells, both of them in culture, there's only about 3% of the mRNAs in the transformed cells that are unique to the transformed state. So that was a surprise when it was first done, but that's not the that's probably not the answer as to, it's not that just cancer genes get turned on. A lot of it is just how much of a thing you're expressing and in what combination of things are you expressing. Okay, here's another H5P question. Okay. Otto Warburg. I know that you've heard about Warburg in Cell and Molecular. I know you heard about Warburg in Dr. Shriver's class in Biochem. But just in case you've evaded that or it's left your brain, he was the first one to document that cancerous tissue has very high rates of glycolysis, up to 200 times faster flux through the glycolytic pathways than in a normal cell. And the end product is lactate. And even in the presence of oxygen, they're still doing lots of glycolysis and still making lots of lactate. This may be due to an alternate isoform of pyruvate kinase that seems to be cancer specific. And so why? Why would you want to do this? Is because then you don't have to depend on delivery of oxygen to grow. You just need the delivery of glucose in order to grow. And if you are drinking through a straw versus drinking through a big bore pipe, in terms of like how much ATP you can get out of glycolysis versus how much you can get out of oxidative phosphorylation, if the stuff coming through the straw is coming out through 200 times faster, you can still get by. So it's like a fire hose going through the straw versus what's coming through the pipe. And obviously it works for cancer cells. So this high glucose metabolism of cancer cells is a marker that you can use actually to determine whether or not is that cancerous cell or not. And they, even in the presence of oxygen, they're doing lots and lots of glycolysis. So one of the things I encountered when I first started doing proteomics, and I don't do a lot of proteomics anymore, but this is maybe 10 years ago when proteomics was new, was that people would publish these papers of, here are the proteins that are made under condition A, control, and here's the proteins made under condition B, some kind of treatment. And whether or not the treatment was cancer, or the treatment was exposure to some kind of heavy metal, or some kind of environmental change, like change in seasonal temperature. No matter what people would find was that enzymes and glycolysis would go up with whatever they were using to stress the cell. And then they would make a story, write a story about in the paper about why that mattered. And really what came out of that was that no matter what is stressing out the cell is going to need more energy. And the common response of that is to do more glycolysis. Glycolytic enzymes are some of the most abundant proteins in the cytoplasm, so they're the most likely you would detect in a proteomics experiment. But it's probably not the thing that is going to be specific to your treatment. But in the case of cancer, high rates of glycolysis are specific to that. Okay. Also, in non-glycolysis, but part of the TCA cycle, there are mutant forms of enzymes that are deemed to be specific to cancer, making metabolites that are only found in cancer cells. And people who do things like metabolomics, like Dr. Shriver, you can use the detection of these metabolites as an indicator of presence of cancer cells. There's even dogs that have been trained to detect lung cancer. That dogs, of course, have a much more sensitive olfactory epithelium than we do, and they can detect volatile components that are given off by people who have lung cancer, and they're pretty good at detecting it. 97% accuracy. Okay, that's it for now, and one more, and then we'll be done with this semester.